Okay, let's unpack this. Imagine one tiny switch inside, well, pretty much every cell in your body. Flick it one way, cells grow, they build stuff, respond to food. Flick it the other way, and they kind of go into cleanup mode, save energy. But what happens if that switch, gets stuck or broken? What if it turns out to be pivotal in, like, everything, how your body grows, how it uses nutrients, right through to big diseases like cancer, diabetes, even how you age? Sounds pretty fundamental, doesn't it? Well, that switch, or maybe more accurately, that whole cellular pathway, is called the mechanistic target of rapamycin, or just mTOR for short. And that's exactly what we're diving deep into today. We've pulled together insights from a whole stack of recent research papers. Our mission here is to really pull out the key pieces, understand this incredibly central cellular, crossroads.
Exactly. That's the goal for this deep dive. We want to take what these sources are telling us and really unpack mTOR's basic functions, and then understand its sometimes surprising links to all these different diseases and aging too, and also look at the potential, which is exciting, but frankly pretty challenging, of actually developing therapies that target it.
Right, and these papers make it super clear. mTOR isn't just like one single entity. It operates in two main forms, these complexes they call mTOR complex 1 or mTORC1.
And mTOR complex 2, mTORC2.
Exactly. And they do slightly different things, but you really need to understand both. So we're going to guide you through what the research says about why this pathway is just so critical. So maybe let's start right at the beginning. What's mTOR actually doing in a healthy cell when things are normal? How does it act like that command center we mentioned?
Yeah, good place to start. Think of mTOR as a really critical sensor. Its main job is constantly monitoring what's going on around the cell. It's basically checking, are conditions good? Are there plenty of nutrients? Are there growth factors signaling it's time to, grow or divide? And when it gets those positive signals, that's when it kicks into gear. Its core function really is linking those good condition signals to actual cell growth, survival, and making new proteins. It's essentially the go signal for building things up. That's what biologists call anabolism. And at the same time, it puts the brakes on breaking things down, which is catabolism. mTORC1 is the main one sensing nutrients and growth factors, driving that protein production and growth. mTORC2 also responds to growth factors, but it's a bit more involved in organizing the cell's internal structure, its cytoskeleton, and making sure survival signals get through properly. It's really all about ensuring your cells build and work efficiently when the resources are there.
Okay, so it's like the cell's construction foreman gives the green light to build when the materials arrive. Makes sense. But here's where it gets, really interesting, and also where the problems can start cropping up. What happens when this system, this foreman, gets stuck? Maybe it's just shouting, build, build, build all the time, even when it shouldn't be.
That's precisely where the research shows these really deep connections to major health problems. The sources consistently highlight a very strong link between mTOR being dysregulated, especially mTORC1 being overactive in metabolic diseases. When mTOR is stuck in that on state, it seems to really mess with how your body handles insulin.
Yeah, it's deeply involved in insulin resistance. The papers point out that chronic mTOR overactivity, and this can be fueled by things like high glucose, high fat levels.
Yeah.
It can disrupt the signaling pathways that insulin normally uses to tell cells, hey, take up sugar from the blood. And this can directly lead to high blood sugar, high blood fats, the classic signs of metabolic trouble, and contribute straight towards conditions like diabetes. And the research gets pretty specific too, doesn't it? It talks about how that continuous mTORC1 activity can make your cells less sensitive to insulin, like the receiver's jammed. And they link this to both type 1 and type 2 diabetes. It affects how cells take up glucose, maybe even how insulin itself is secreted. And maybe not surprisingly, the papers explicitly say that even just eating a lot of sugar can directly flip on this mTOR pathway.
And the connection definitely extends to obesity, too. Overactive mTORC1 is linked with increased triglycerides, fat in the blood, and also reduced breakdown of fat, so it contributes to fat accumulating. They also draw a line to non-alcoholic fatty liver disease, or NFLD. They point to finding higher levels of certain proteins that mTORC1 controls in the blood of people with NFLD. It really paints this picture where an overactive mTOR is basically telling the body, store fat, don't break it down, even when that's harmful.
Okay, now let's switch gears a bit. Another huge area where mTOR seems to be a key player is cancer. I mean, cancer cells are basically cells that have lost control, right? They're just focused on growing and dividing like crazy. So it makes a kind of intuitive sense they'd want to hijack a pathway like mTOR, which is all about promoting growth.
Absolutely. And the research shows it's not just intuition, it's really happening. The sources suggest the mTOR pathway is somehow exploited in roughly 30% of all cancers.
30%? Wow.
Yeah, and in some specific types, like breast cancer, that number jumps much higher. Some studies find it's dysregulated in around 70% of breast cancer cases. So when cancer cells manage to crank up MTO activity, they're giving themselves this massive boost. It ramps up the production of proteins they need for super fast growth, for moving to new places, metastasis, and even for building new blood vessels to feed the tumor, angiogenesis. Cancer cells need tons of energy and building blocks, right? And MTO helps them get it by messing with metabolism, like pushing this process called aerobic glycolysis. It's a fast, maybe less efficient way to burn sugar for energy, even when there's oxygen around.
Okay, here's something from the sources that really surprised me, though. mTOR normally inhibits something called autophagy. And autophagy is basically the cell's cleanup crew, its recycling system. It breaks down old, damaged parts.
Right. And what's fascinating, and maybe a bit counterintuitive, is that the papers highlight that a lack of effective autophagy is actually linked to cancer developing.
Yeah.
So mTOR, by suppressing this essential cleanup process, it might actually inadvertently help tumors get started, because the cell isn't efficiently clearing out damaged components that could potentially become cancerous. It's like disabling the garbage disposal. Junk builds up.
And the research gives specific examples, too. While mTORC1 is often the main culprit, mTORC2 also plays a role, especially in certain cancers, like prostate cancer, apparently. It often gets activated there when a really important tumor suppressor gene, a gene meant to stop cancer, gets lost or mutated.
But it's not just a one-way street, mTOR promoting cancer. The sources also mention things that can put the brakes on mTOR. These can act as tumor suppressors themselves. One example they give is a protein called NLRC3. The papers suggest levels of this protein are lower in colorectal cancer. And here's where it gets really interesting. Again, they touch on this idea that even things outside the cell, like your diet, or even the specific types of bacteria living in your gut, these might influence colorectal cancer risk by affecting mTOR signaling inside your cells. It directly connects our lifestyle choices to this internal pathway.
Okay, moving on then. One of the most, I think, exciting and widely talked about areas recently is this connection between mTOR and aging. And what seems truly remarkable reading these sources is how fundamental this link seems to be. It's conserved across just a huge range of life, from simple yeast all the way to mammals, like us.
Yeah, the evidence across different species is really quite striking. Study after study shows that if you dampen down mTOR activity, whether you do it genetically or through interventions people talk about like calorie restriction, it consistently extends lifespan in many different organisms. The papers cite specific examples like genetically tweaked mice that have reduced mTOR activity, living significantly longer lives.
And this connects right back to that autophagy thing, the cellular cleanup crew we were just talking about, doesn't it?
Exactly. Inhibiting mTORC1 seems to be a key way to promote autophagy.
Right. And this whole idea of cells being better able to clean themselves up, get rid of damaged junk, that's seen as a really crucial mechanism for healthier aging, maybe longer life. It seems especially relevant because we know autophagy tends to decline naturally as we get older. And remember, mTOR normally keeps it suppressed.
The research also links mTOR to senescent cells. You know, those old cells that stop dividing but just hang around?
Yeah, the zombie cells, people call them sometimes. They cause inflammation.
Right. They contribute to age-related decline. And the papers suggest mTOR signaling can be kind of stuck on in these senescent cells. And maybe, just maybe, targeting mTOR could be a way to help clear these cells out. They even touch on how mTOR influences things like mitochondrial function, the cell's power plants, and even shifts in immune function, all things that change with age and seem tied into this pathway.
So given everything we've discussed, mTOR's central role in growth, metabolism, cancer, aging, it seems like such an obvious target for therapies, right? Therapies aimed at treating diseases or maybe even influencing the aging process itself. What have researchers actually developed to try and hit this target?
You're absolutely right. It's a major, major focus of research. And scientists have developed different generations of inhibitors. The first generation really started with the discovery of rapamycin, found in soil bacteria on Easter Island, believe it or not. So you have rapamycin itself and drugs derived from it, often called rapalogs. Now, these first-generation drugs, they mostly target mTORC1. But, as the sources point out, they have limitations. For one, they don't completely shut the pathway down. It's more like turning down the volume, not switching it off. And sometimes that partial inhibition can actually cause the cell to compensate by ramping up other related growth pathways, which can make the drug less effective or lead to resistance. Plus, they definitely come with side effects, things like lung problems, potential fertility issues, and worryingly, some studies link them to causing new onset diabetes in some patients.
Okay, so those limitations led to the next step, right?
Yeah.
The second generation inhibitors. What's different about those?
These were designed to be more comprehensive, basically. They're called ATP competitive inhibitors. They work by blocking the energy source, the ATP, that mTOR needs to function. The idea is to inhibit both mTORC1 and mTORC2 more completely. The papers mention drugs like sapanisertib as examples, and they are showing some promising results in certain cancers, sometimes especially when used in combination with other treatments, maybe drugs targeting tumor blood vessels, or even, quite interestingly, with the common diabetes drug metformin. Metformin itself seems to have some mTOR inhibiting effects.
And the research doesn't stop there, right? People are looking at even more sophisticated ways to target mTOR.
Exactly. There's talk of an emerging third generation of inhibitors. These are being designed to target multiple parts of the mTOR protein complex itself, trying to be even more specific or effective. The sources briefly mention one called Rapalink-1, showing promise maybe in organ transplantation, potentially being better at suppressing the immune cells that cause rejection. It just highlights that we're constantly trying to refine how we approach this incredibly complex target. And also exploring if existing drugs like metformin or even natural compounds like taurine might have useful mTOR effects in fighting diseases like diabetes or cancer.
But while all this therapeutic potential sounds incredibly exciting, the sources are also really clear that targeting mTOR is, well, it's not simple. The pathway's complexity means we sometimes see unexpected results, or things aren't quite as straightforward as just inhibit mTOR, fix problem.
Precisely. Take mTORC2, for example. Its role seems more nuanced, especially when you look at aging and metabolic health. While generally turning down mTOR might seem good, the chronic use of some of those first-generation drugs, which can eventually disrupt mTORC2 function, well, that's been linked to some of those metabolic problems we mentioned earlier, like contributing to diabetes. It's complicated. And remember that autophagy point we discussed, how inhibiting mTORC1 boosts it.
Yeah, the cellular cleanup.
Right. Usually seen as a good thing, especially for clearing damage and maybe suppressing tumors. But the papers also note that in some specific cancer situations, cancer cells can actually hijack autophagy to help themselves survive when stressed, so it's not always beneficial.
All right, the cancer cells find a workaround, they adapt. And even with newer, more specific inhibitors, cancer cells are just notoriously adaptable. The sources mention cases where you block mTOR, and the cancer cells just find other metabolic pathways to rely on for energy and growth. They develop resistance. Plus, you're always walking a fine line, aren't you? Inhibiting a pathway as fundamental as mTOR, which normal, healthy cells absolutely need, always carries that risk of toxicity or unwanted side effects.
So when we sort of pull back and look at the whole picture from these research papers, what's the main takeaway? I think the core message is really undeniable. mTOR is a true master regulator inside our cells. It acts as this critical junction box, linking the outside world, the food signals, the growth signals, to really fundamental internal processes. Growth, how we use energy, survival itself. And when it goes wrong, when it's dysregulated, it's clearly a central player in so many of the major diseases we grapple with today. Metabolic disorders, a huge chunk of cancers, and definitely the aging process.
It really drives home that idea of balance, doesn't it? Too much mTOR activity seems problematic, driving disease, maybe speeding up aging. But then carefully reducing its activity, if done the right way, shows such enormous potential for extending our healthy years, our health span, and for developing new ways to treat really serious illnesses.
Absolutely. And even with all the complexities, all the challenges in designing therapies that are specific and safe, the sheer amount of ongoing research detailed in these sources, the development of newer, more targeted inhibitors, the exploration of combination therapies, it all continues to make mTOR a profoundly exciting and, I think, genuinely promising area for future health breakthroughs. It really feels like we're still just scratching the surface of what manipulating this pathway could achieve.
So, maybe as you go about your day-to-day, just take a second to think about that incredibly intricate web of signals firing inside you, processed by pathways like mTOR. Everything from the food you choose to eat to the stresses your cells might be under. Understanding this single cellular crossroads could, maybe, unlock truly profound new ways to influence our health and how long we live well in the years ahead.
Indeed. You know, what possibilities does this deep dive really open up? When you think about personalized medicine, maybe tailoring treatments based on an individual's mTOR activity, or even just rethinking the impact of the lifestyle choices you make every single day, it's definitely something to ponder as you navigate your own internal cellular landscape.